Our next talk is by Joanna Wardwell-Ozgo from Emory University. And the title of her talk is A Kinome-wide RNAi Screen in Drosophila Glia and Human GBM Models Reveals Stk17A Drives Neoplastic Glial Proliferation.
I'd like to begin by thanking the organizers of this session for allowing me the opportunity to speak to you about the work that I've been doing as a postdoc in Renee Reed's lab, defining the mechanism of a novel kinase that we identified in a kinome-wide RNAi screen and its involvement in glioblastoma. In the Reed lab, we're interested in studying glioblastoma, which is the malignant version of a class of brain tumors called gliomas. Glioblastoma is the most common and deadly CNS tumor. It's proliferative, invasive, and resistant to all current therapies. The most frequent genetic perturbations found in patients show frequent mutations that allow for the constitutive activity of both EGFR and the PI3 kinase pathways. Unfortunately, targeted therapies against different components of these two pathways have not been successful in the clinic, which illustrates the need to both better understand the complex biology behind this disease, as well as isolate potential new therapeutic targets. With this in mind, we created a novel model for glioblastoma in Drosophila. This is due largely in part to the conserved function and biology of glia from Drosophila to humans. And we do this by chronically activating constitutively active alleles of EGFR and PI3 kinase in the glia of the brain. And what you can see here is a wild-type third instar larval brain, the nerve cord in the central brain. And we use RepoGal4, which is a driver that expresses in all glial populations to drive EGFR and PI3 kinase signaling. We get a gross overproliferation of these cells. These mutant glia are proliferative, invasive, and recapitulate some of the characteristics of the human disease. With this model in hand, we performed a genetic modifier screen across the entire kinome by comparing the neoplastic phenotype to the neoplastic phenotype with the loss of a kinase via RNAi. This allowed us to ask the question if any other kinase suppressed or enhanced our neoplastic phenotype. This screening approach revealed DRAC as a novel kinase involved as a suppressor of the neoplastic glia. What we do know about DRAC comes from a developmental context, where in 2010, Hiffner's group described that DRAC interacts in concert with and semi-redundantly with ROCK to phosphorylate spaghetti squash or squish. A few years later, Frank Pichard's group then went on to describe that it works downstream of EGFR and Rho signaling to phosphorylate squish during morphogenesis. Now, DRAC's direct phosphorylation of squish allows for modulations of the cytoskeletal dynamics, which result in changes in migration, cytokinesis, apoptosis, and proliferation, all of which are important for a cancer cell. So here is an image of a third instar wild-type larval brain hemisphere. You'll see outlined in green are the glial cell bodies. In red are the glial nuclei, and in blue are the neural projections. When we overexpress EGFR and PI3 kinase in these glial cells, we get a gross overproliferation of the glia. And when we knock down DRAC in this mutant glial system, we see a strong suppression of the phenotype. It's important to note that knockdown of DRAC in a wild-type setting does not change any phenotype and architecture of the brain, illustrating that the neoplastic state is particularly sensitive to levels of DRAC. Since we knew upstream and downstream components of DRAC, we next sought out to determine if those components also suppressed the phenotype. And indeed they did. When we used RNAi against RHO-A, we saw strong suppression of our neoplastic phenotype. Similarly, when we also used RNAi against spaghetti squash, we saw a strong suppression of our neoplastic phenotype, illustrating that DRAC and its pathway components are important. Also, interestingly, when we knocked down ROC, we saw no genetic interaction. So I wanted to confirm that spaghetti squash was the important downstream target of DRAC, and I did this by modulating levels of flapwing, which is the phosphatase that removes the phosphorylated sites, or the phosphorylation of squish, and returns it back to its non-active state. When you look at the mutant glia model with flapwing also overexpressed, we see a strong suppression of the overall phenotype seen here. Also, when we knock down flapwing, removing the regulation of squish, we see an enhancement of our phenotype, illustrating that squish is an important downstream target of DRAC. Changes in the phosphorylation of DRAC can lead to multiple different things, and we wanted to determine which was important and for DRAC's phenotype. And luckily for us, a few months ago, Jeffrey Thomas's group published a paper that hinted that DRAC's phosphorylation of squish might be important for cytokinesis. So his group found that DRAC's phosphorylation of squish was necessary for nilin, which is a scaffolding protein that organizes microfilaments into the contractile ring, which is important during cellularization and cytokinesis. So DRAC's direct phosphorylation of squish was important for nilin to bind to myosin and to organize it into a contractile ring for cytokinesis. So I asked what happened when we knocked anilin down in our mutant glial background, and when we did that, we saw a strong suppression of our phenotype, illustrating that this proper organization of the microfilament ring in cytokinesis is likely important, and how DRAC is eliciting its effects. The Reed Lab is also very translationally focused, and so part of my attention has also been to determine whether or not DRAC is important for human cancers. So the human ortholog of DRAC is called Stk17A, and it phosphorylates MRLC, the ortholog of squish. And when we look across a panel of human tumor, glioblastoma tumor stem cell cultures, we can see varying levels of squish in comparison to the control. Importantly, these levels also coordinate with levels of mutant EGFR status, and EGFR here is constitutively active. Also, I'd like to draw your attention to the fact that when squish is when Stk17A is overexpressed, we also see an increase in the diphosphorylated active version of MRLC. So we saw this in tissue culture. I also wanted to determine whether or not this had any real implications in actual tumors. And we did this a few ways. By looking through TGCA data sets, we can see that squish has a copy gain alteration in about 75%, 76% of all glioblastomas, and it's overexpressed in about half. Interestingly, Stk17A is also altered in the lower grade gliomas. It's overexpressed in about 20%, and it has a copy gain alteration in 23%. This really illustrates that Stk17A might be a driver of the neoplastic transformation of glial cells into cancer. So using IHC to stain for Stk17A in tissue samples, we can compare the normal brain, which has very little, if any, Stk17A staining, to glioblastoma patient sample. And you can see strong staining of glioblastoma, or Stk17A in brown in the glioblastoma patient. In a lower grade glioma, in a grade 3, we can also see strong Stk17A. And interestingly and importantly, this also, in the same patient, you can see strong staining of phospho-MRLC, showing that we can see upregulation of Stk17A in actual patient samples, and that it does seem to correlate with levels of MRLC. Our fly data illustrated that Stk17A might be exerting its effects by changing cytokinesis, so I wanted to determine whether or not the proliferation of glioblastoma cells was affected with Stk levels. To do this, we used shRNA to knock down Stk17A in GBM cells. And when we knock down Stk17A in GBM cells, we also lose the diphosphorylated version, the active version of MRLC. And when we assay for proliferation, compared to control, loss of Stk17A has a dramatic and significant effect on the proliferative rates of these cells, illustrating that Stk17A is important for the proliferation of GBM cultured cells. Altogether, today I hope I've shown you that DRAC, a novel kinase that we identified from a kinome-wide genetic modifier screen, is a rate-limiting step for EGFR PI3 kinase-driven neoplasia. We believe that it enacts its effects by increasing the rate of cytokinesis after mitosis and allowing the cell cycle to move forward. STK is also found in human cancers, both in tissue culture, allowing us to study it, as well as human patients. And proliferation and Stk17A knockdown decreases the proliferative rates of these cells. We hope that Stk17A then now stands as a therapeutic potential target. And those experiments are to be completed or actively being pursued. I'd like to thank everybody in the Reed Lab, especially Renee, for her help and guidance in this project, and Nilong and Deidre, who are undergraduates who have helped me complete this work, as well as Colleen and Jomar. Our collaborators, both at Emory, UCLA, and USCD, as well as the funding sources to Renee that allowed this project to happen. I'd like to thank the GSA for giving me a travel award to come talk to you about my research, as well as the first program at Emory, which is an IRACTA Career Development Award, of which I am a fellow. I'd be happy to take your questions. Hi.
All right, yeah. You say that when you knock down your system in wild-type glial cells, there's no phenotype. But do you know if this is necessary for neurons or any other tissues, or could it be a potential target because it's not needed in other tissues?
I don't believe. So when Renee initially isolated this kinase from her screen, she did the expression patterns in different tissue types in the brain, and changes in the neurons did not elicit any type of effect.
Okay, and another very quick question. At the beginning, you showed that this reduces clearly the size of the brain, but if I'm right, I saw a lot of nuclei. So it's clear that at least the membrane of the glial cells is reduced. But have you counted the nuclei in the flies? The number of nuclei?
No, not yet, and that's something that we're definitely interested in pursuing, especially since we do think that it is involved somehow in proliferation. You're right; it is something that we need to take care of.
All right, thank you.
Thank you.
Hi. Is this on? Yeah. Glial cells are post-mitotic, right? So when you overexpress EGFR and other components, so what you see is overproliferation. Do you think that it converts back to a stem cell-like neuroglioblastoma and then divides, or it's just glial cells dividing abnormally?
So I believe if you're asking, you're saying that glial cells are post-mitotic and overexpression of EGFR and PI3 kinase, is it kind of reverting it to a more stem-like phenotype?
Yes.
Is that what your question is?
Yes.
It's definitely driving proliferation through reactivation of the cell cycle. So when you look at different cell cycle markers, you're seeing an increase in all the classic cyclins and all that. I don't believe that they're actually de-differentiating into their precursor cells.
Have you looked at any neuroblast-specific markers?
For the model of our glioblastoma, yes, yes, and only expressing in the glia and expressing EGFR and PI3 kinase give us this effect. Wonderful, thank you.
Don't go away.
Oh, I'm sorry.
We have a little time, and I had a question. Have you looked at all at migration, or have you started just with the cytokinesis?
I did look at migration, and the results weren't exactly encouraging or obvious, and so when that paper popped up about DRAC's importance for cytokinesis, it was kind of encouraging.
So along the same lines, you saw variability in the Stk17 levels. Is there any correlation with them being more metastatic?
So these cells aren't necessarily metastatic, but they are invasive.
Okay,
And right, they definitely, EGFR amplification and Stk17A in a few of the cell lines that we've looked at have shown to be migratory in the brain itself. Thank you.